We will have a fruitful discussion, first by 6 presentations, followed by a moderated discussion on the presented subjects and presented science. Unfortunately, I cannot see the first speaker, Dvir. Is he or she present? So that does not seem to be the case. In this case, I will continue with my own presentation, and then we will see if this person does arrive, and we can then switch back. So therefore, I like to share my screen, or is there Dr. Dvir present or not? I think not present. So then I will share my screen. Okay, so ladies and gentlemen, I'm going to talk about imaging and image analysis for stem cell mediated distraction osteogenesis. This is not my only work. It is an interdisciplinary work, and I will mention the different contributors during my talk. I am a professor for material science and medicine at the University of Basel, and my background is physics, as you might have seen in the CVs. We are doing high-resolution hard X-ray imaging in our department, and we have a core facility on micro and nanotomography. And for many years, we are dealing for this technique that is used for different purposes. For example, to precisely measure the precision of oral scanners or maybe aligners. We are also investigating unique objects. For example, you see here an example from the Natural History Museum in Basel, where we can visualize internal structures within opaque unique objects. And the same applies, for example, for the anatomy of surgeon organs or tissue-engineered constructs. And in addition, we do a lot of technical developments with lab-based instruments. For example, we have integrated an grating-based interferometer into our nanotome, and we are part of synchrotron radiation-based microcomputer tomography, for example, the anatomics beamline at the Synchrotron Soleil in France. And what I like to show here is how we can take advantage of micro and nanocomputer tomography for osteogenesis. And what we are going to present here is combining the micro and nanotomography and histology to characterize this distraction osteogenesis in rats. And I think many of you are familiar with that, maybe better than I am, because my background is physics. But these experiments were planned and carried out by craniomaxillofacial surgeons, Christoph Kunz and Claude Jacqueri. And what they have done, they have taken rats, they have cut the bone. There was a so-called latency duration of about 5 days. And then the distraction took place with a certain device I'm going to show on the next slide in more detail. And then it was followed by a consolidation phase, the late consolidation, and finally the remodeling. So we have really produced newly formed bone that forms here and gives rise to a better functionality. And what had been proposed in literature is that the stem cells should be injected after the latency phase, so after this phase given here. What was proposed by Christoph Kunz was that one should inject the stem cells already before this phase, that should be more effective. And therefore, we made the hypothesis that stem cell injection at an early stage of distraction improves the clinical outcome. And the second hypothesis is more related to research in my team, and that is post-mortem three-dimensional imaging complements the well-established histological analysis. You see that such a distraction device has to be designed for the rats, so not for the humans, but for the rats. So they are much, much smaller, and for sure they have to be adapted to the anatomy of the rats. And you see here this example that was produced by a company and had different parts that are helpful to perform that kind of intervention. The result can be seen here at that three-dimensional representation that was generated by Georg Schulz on the basis of conventional microcomputed tomography. And what you can clearly see here is the newly formed bone. You see it here, and you see also the holes from the screws from the distractor. So therefore, we get quite a good idea how it looks like. But for the histology, we need to decalcify the tissue, and therefore the bony structures as well as the teeth are not seen as well as before. And for that purpose, Christine Tanner, a computational scientist, and Guido Sigron, also a craniomaxillofacial surgeon, they have worked together to identify landmark pairs. That means pairs that comes from the non-decalcified tissue visualized with our laboratory-based microcomputed tomography and the decalcified tissue that we have visualized at the synchrotron radiation facility in Hamburg at the Doris storage ring. And what you can clearly see here is a tooth cut in the 3 orthogonal directions. And Christine has looked for the landmarks in the non-decalcified and decalcified tissue. And you see that it is relatively easy to find them. For sure, one has to validate it. And that had been done by Guido Sigron here, for example, at a lacuna in the newly formed bone. You see here the three-dimensional distribution of the landmarks that had been used for registration, and the bold lines here correspond to the examples given on the right-hand side. What one can easily recognize is that a rigid and scaling approach is inappropriate because we have non-rigid, that means deformations that are local. And we have shown here the rigid registration and here the non-rigid one. And from the non-rigid one can derive the local shrinkages that are presented here with the color bar. So by the preparation procedure, we have a certain shrinkage, and this shrinkage is substantially, especially present here at the bone that is more mature. So therefore, we can say the decalcification results in a shrinkage that could be quantified. One can even see it better by comparing the rigid registration with the non-rigid one. And you see here the lines on the right that are given in orange does fit much better than the lines given here in turquoise for the rigid registration. And therefore, the non-rigid registration that is not so easy is very important. And if one does the registration in a perfect fashion, then one can produce the so-called joint histogram or the bivariant histograms that combines the X-ray absorption from the synchrotron radiation-based data with the X-ray attenuation we have found from the data of a non-decalcified tissue within our lab source, nanotome. So the nanotome you see here the histogram or part of the histogram, and you see here the histogram of a slice I will show you later on. The same you see for the decalcified tissues, where we see several peaks, so not just the peak of the bone and the peak that is related to the surrounding air, but you see more structures that are present there. Now, the slice I mentioned is this one. So this is an H&E stained slice. And for sure, one can try to identify the position of a slice in the three-dimensional data from the lab source and the three-dimensional data from the synchrotron radiation facility. You can see some of the similarities by eye, but for sure, we want to have better than this manual registration. And for that purpose, we have developed a pipeline for the automatic refinement of the position that had been done by my team. And you see here the pairs of landmarks in the synchrotron radiation-based microtomography data and in the histology data. And you see they fit quite well. The next step we have done is the analysis of a common area for the three imaging modalities individually. That means we have just taken, for example, this slice, from this virtual slice from the lab-based microcomputer tomography, and you see that we can discriminate between the air, that means the surrounding, and the bony tissue. And you see here that we do see the bony tissue, that means the major part here, and the one formed during the distraction. Here you see the same slice position in the synchrotron radiation-based micro-CT data, and you see that we can discriminate within this histogram different areas. So we have the air again that is made transparent, and then we have a black, a green, a red-colored and a blue-colored region, and here we have applied the so-called k-means clustering. The same can be applied for the histology slice, but because we have different color channels, we can do it in a different fashion. So therefore, we have a kind of overlapping in these k-means, and we can recolor the slice accordingly. And what you realize is that there are some similarities. So the major bone here can be identified, and we see some other structures with some similarities. So therefore, it's worth trying to combine techniques. And in the first step, we have combined two of the techniques each. So for example, here you see something that I have shown you already before, but not for the individual slice, but for the full 3D data set. Here you see the combination of the micro-CT in the lab and the histology. And here you see the combination of the synchrotron radiation-based micro-CT and the intensity that comes from the histology. And then you can produce again these structures, and you see that we reach a better similarity that is not surprising, but might be helpful. For sure, the best is to produce a trivariant, not just a bivariant histogram, but a trivariant histogram. And this is shown on this slide. So what you see here is that we combine the microtomography data before decalcification, the microtomography data obtained at the synchrotron radiation facility after decalcification, and then the two-dimensional histology that is more or less standard. We put them together in this joint histogram, or we say also the trivariant histogram, because we have three modalities where we combine it. And by the k-means clustering, we can produce an image, as you see here, where we can discriminate different kinds of tissue. So we have soft tissues with many nuclei given in black, the bone, and the major bone mainly, that is given here in blue. And you see also a part of that bone that is newly formed. Then we have connected tissue with fat given in green, and the newly formed bone, that is really the new formed bone, is given in red. So therefore, we can conclude that multimodal imaging and registration support the identification of tissues. And Guido Siggon, who was one of the driving persons in that study, has asked the question, can we even identify different types of collagen just by that trivariant histogram analysis? And the preliminary data indicate that it is beneficial to have a stem cell injection at an early stage of the distraction. And for sure, the question occurs, can we see these human stem cells within the tissue of a rat generated? And that was investigated by the team of Arno Scherberich from the University Hospital in Basel. And Arno has organized different staining. So for example, the human specific genomic ALO sequences does help to identify the human cells. And these are here given in blue. This is a zoom in from a certain area here. And he has also done the immunostaining for HLA1, a human-specific marker. And you see it here in red with this part, a zoom in from that one. So we can really say that there are osteocytes present. So with that, I like to summarize. And I like to tell you that preliminary animal studies were done. These studies have included 4 animals, but our analysis, I have shown, is just restricted to a single rat because there was an incomplete osseous bridging in the other 3 animals. So you see, it's not always simple to perform such studies. The single rat is a basis of that Dvir study. And I believe I was able to show you that it is worth combining microcomputed tomography, that is a 3-dimensional method, with histology, that is mainly a 2-dimensional method. For sure, we can make serial sections and get a 3D representation out of it. Non-registration and the extraction of common volumes of the common areas in 2D allow for segmenting anatomical features. And we hypothesize here that combining laboratory-based micro-CT 4D calcification, synchrotron radiation-based tomography after decalcification, and histology with HA stain can support the discrimination between the types of collagen. The injected human mesenchymal stem cells survived and differentiated into human osteocytes. I think this is also an important protocol that has been established by the team of Arno Scherberich. And to quantify the impact on injected stem cells at the beginning of a distraction phase, we need animal studies with a sufficiently large number of rats, and that has to be performed. Let me give a kind of summary highlighting that this is really an interdisciplinary endeavor. So that means we have to collaborate. For sample preparation, we do need surgeons, for example, experts in life sciences, to prepare the unique objects, and we want to avoid the physical slicing, and therefore we are looking for X-ray imaging in 3D. Staining or related techniques are usually very helpful and have also to be performed by the life science specialists. The data acquisition is usually done by a team that is very often headed by physicists, in some cases by engineers. So therefore, we start with laboratory equipment that is easily accessible, and then we can go to the synchrotron radiation facility where we have to apply for beam time and usually need quite a team for the data acquisition that takes place 24 hours, 7 days a week. The data reconstruction is usually done by physicists because they can stitch the data, they can retrieve the phase if we are looking for phase contrast. Sometimes we rely on on-site reconstruction, but artifact removal is a part of that work. The visualization of the data is usually done by computer vision experts, and here we usually apply available softwares, but the data sets are huge, and sometimes the software does not work properly. Data quantification is usually done by computer scientists. So they perform segmentation, as I have shown you, registration, I have shown you. But in addition, they use other tools like the distance transform or the active contour models. And finally, we have to combine the data, as I have shown you, for example, the histology data, usually obtained by pathologists, for example, the registration and the multivariance histogram. And here we need a wide spectrum of scientists. And you see here this example. We are going to publish these results, and the manuscript is now for the proofreading. So I hope you can see it soon. Thank you very much. And now we come to the next presentation that will be given by Letizia Nicoletti. And Letizia, can you share your screen, please? Yes.
Yes.
Maybe you can switch to the other mode that we see. Yes. 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 Now it's perfect. So please start with your presentation.
Okay. Thank you, and I would like to thank the organizer for giving me the opportunity to present this talk based on novel microRNA-releasing lipoplexes, encapsulated non-injectable hydrogel for cardiac regenerative medicine. Cardiovascular diseases are the leading cause of mortality worldwide. Among these pathologies, myocardial infarction is responsible for 85% of death due to the inability of the adult human heart to regenerate after injury. Myocardial infarction causes a decreases or interruption of blood flow and oxygen to the cardiac muscle, with the formation of a fibrotic scar, mainly populated by non-contractile cells. So there is an urgent need to regenerate myocardial tissue after ischemic diseases. Heart transplantation is the only therapeutic solution for restoring the damaged functionality tissue, but it has great limitation. New technologies based on regenerative medicine solutions are investigated, including cell-based therapies, biomaterials such as scaffold, hydrogel and patches, and gene therapy. The local release of microRNA is a promising strategies for cardiac regeneration. But the direct administration of microRNA is limited by poor cellular internalization, short circulation time, and off-target effects. So for this reason, viral vectors are widely used to deliver genetic material, but their clinical application is limited by different drawbacks. New therapeutic solutions based on nanoparticles are exploited as the safer approach and for easy of production and low cost. This work is part of the BioRecard project, which is an ERC-funded project which propose a new regenerative medicine approach to recover myocardial functionality contractility in the post-infarct fibrotic tissue. The BioRecard project includes 3 goals. Specifically, I work on the first task based on the design of a multifunctional hydrogel for direct reprogramming in vivo. Our group has recently demonstrated that a combination of 4 microRNA called MIR combo can trigger the transdifferentiation of adult human cardiac fibroblasts into induced cardiomyocytes, which was previously demonstrated in mouse model. So the aim of the work was to design new microRNA-releasing lipoplexes and the development of an injectable hydrogel for in situ release of microRNA-loaded lipoplexes. Firstly, new microRNA-releasing lipoplexes were developed, providing a high encapsulation of the oligonucleotide, a gradual release of microRNA, and a high transfection efficiency of adult human cardiac fibroblasts. New lipoplexes loaded with NEGMIR as negative control or MIR combo, based on a mixture of cationic and helper lipid, were prepared at different amino to phosphate ratio called NP ratio, thanks to electrostatic spontaneous interaction between the cationic lipid and the microRNA negatively charged. NEGMIR-loaded lipoplexes were physicochemically characterized by dynamic light scattering. As we can see from this figure, a decrease in hydrodynamic diameter and an increase in zeta potential from negative to positive values, increasing the NP ratio, were obtained. Based on these results, the NP3 was selected for further experiment aimed at comparing their physicochemical characteristic and biological effect with those of commercial transfection agent, the Dharma-Fakmirna lipoplexes. NEGMIR-loaded lipoplexes had a high encapsulation efficiency, about 99%, compared to Dharma-Fakmirna lipoplexes, with a gradual and total release of the microRNA for lipoplexes compared to Dharma-Fakmirna lipoplexes. In vitro validation studies were also performed in adult human cardiac fibroblasts, showing a high biocompatibility and the ability to efficiently internalize the FEM-labeled MIR1 into cells. Moreover, a high level of MIR1 was obtained with lipoplexes compared to Dharma-Fakmirna lipoplexes. Moreover, the transfection efficiency was also evaluated, showing a TVF1 downregulation. Finally, MIR combo-loaded lipoplexes were developed and physicochemically characterized. The size, the potential, and encapsulation efficiency were similar to NEGMIR-loaded lipoplexes. After 15 days post-transfection, a tenfold increase of TNNT2 cardiomyocytes marker was observed, and also a high percentage of cardiac troponin positive cells was obtained for lipoplexes compared to Dharma-Fakmirna lipoplexes. Overall, these results are promising because we could improve the direct reprogramming only by changing the type of nanocarrier with the same oligonucleotide. The second aim of the work was the development of an injectable hydrogel for in situ release of microRNA-loaded lipoplexes. Alginate-based injectable hydrogel are investigated in clinical trials to favor cardiac tissue remodeling after myocardial infarction as Algicil L2R. However, alginate have some limitation, such as the enzymatic degradation in human body and the poor cell adhesion. To overcome this limitation, we oxidized alginate, obtaining alginate dialdehyde hydrogel. The alginate dialdehyde hydrogel have different advantages, such as faster degradation rate, low molecular weight, and free aldehyde group that can form covalent bonds with biomolecules. We obtain a degree of oxidation of 25% and a yield of production of 70%. The ADA hydrogel was formulated through ionic linking using calcium ions through double siring mechanism. ADA hydrogel composition was rheologically optimized, varying alginate and ADA ratio in order to obtain similar viscoelastic properties closer to Algicil product. Finally, the lipoplexes were loaded with silencing RNA, loaded with Chinin-5. And they were physically encapsulated into the selected ADA hydrogel. As we can see from these results, rheological properties of hydrogel loaded with silencing RNA lipoplexes maintained their rheological properties, and we can see a fast release of the microRNA until 24 hours. This result suggests that lipoplexes can disaggregate in the hydrogel due to a possible interaction, electrostatic interaction with the compound of the hydrogel. So based on this result, we design new hybrid nanocarriers. In collaboration with University of Turin, with Professor Arpico and Stella, we patent a new technology based on coating of lipoplexes that improve the release of microRNA up to 28 days. Now we have the direct reprogramming tests are in progress. So in conclusion, new lipoplexes based on a mixture of cationic and helper lipid were developed, allow more efficient microRNA delivery to adult human cardiac fibroblasts compared to Darmafect, and enhance direct reprogramming. Moreover, the design of alginate ADA-based hydrogel were developed for delivery of microRNA-loaded lipoplexes with mechanical properties close to Algicil L2R. Current studies are in progress based on the exploitation of other groups for functionalization with biomolecules, and also the exploitation of the patented technologies for direct reprogramming. I would like to thank my supervisor, Professor Valeria Chiono, and Dr. Elena Marcello, and Camilla Poletti. And I also would like to thank all of you for your attention.
So thank you very much. A lot of female power here, I see.
Yes.
I think it's very typical for Italy. I just came back from Pisa, so I have seen it there as well.
Yes.
But very interesting, and I think we will have a fruitful discussion about these lipoplexes and these nanostructures that you're using for preclinical purposes. Okay. Thank you also for keeping in time. That is very, very helpful. So we are perfectly in time. And now we can switch to the last presentation within our session. And that will be given by Sammy. So I think everybody can read the title perfectly well. And I leave the floor for this presentation.
Okay, hello. Thank you for this kind introduction. I'm Sammy. And first of all, I want to thank the Kleinam for this opportunity to talk here. And I want to talk today about quite known, meanwhile, technology in science, 3D printing and 3D bioprinting, and how we apply it to design in vitro systems to fight against bacterial infections with nanoantibiotics. And I want to start firstly with another quite recent topic in science, the human microbiome. Which is basically the aggregate of all your microbes within human bodies. And as any body tissue or body part, also hair follicles and the skin, the largest organ, have their own microbiome. And when there is now a shift in the diversity or the balance of the microbiome towards kind of rather bad bacteria, like a popular example is Staphylococcus aureus, this can lead to severe inflammation processes, with the consequence of diseases like acne inversa or folliculitis decalvans, with awful lifelong scars. And the ability of many of these strains to form biofilms exacerbate the whole situation with regard to therapies. So the medical and scientific community are working and developing novel anti-infectives to fight these infections and biofilms. And they figured out also nice and innovative tools to accurately targeted at the spot of infection via nanocarriers and nanoparticles, because it was shown in literature that nanoparticles can enter with the hair follicle openings and really work there at the spot of infection with loaded drugs and fight the infection. But as every other formulations, all kind of formulations have to be tested on suitable platforms. And from our point of view, we think that, for example, animal models are not suitable due to genetic, immunological, and anatomical differences, also ethical, of course. However, when we look on the in vitro alternatives, we see there is somehow a limitation of hair follicle models, because currently hair follicle models are limited to isolated human but still living hair follicles swimming in the medium, basically, which is not really as a model suitable for topical transport systems like nanocarriers to be tested. So our basic idea is to design a 3D organ culture of hair follicles as a model, which is supposed to be applied for nanoparticulate testing, where the hair follicles isolated from humans and still living are inserted into a dermis substitute, and it's then supposed to be tested with nanoparticles, and the readouts will be follicular transport, but also the interaction and the reaction of the hair follicles upon a treatment or the induction of an inflammation. So that's why it's important and pivotal that the hair follicles in there are living. So the first step towards such a model was to collaborate with a local cosmetic surgeon here in our town, Saarbrücken, and from who we received haired skin tissue from facelifts. So what we do is then we remove the hair follicles from this skin tissue by removing the epidermis and most of the dermis. And then we pull out the hair follicles with fine tweezers, we collect them and make a quick quality control. And then what we wanted to do before we go to the 3D environment, we wanted to see whether we can culture the hair follicles in a conventional method with all our medias, etc. And indeed we were. Our primary readouts were the hair growth and the morphology. We could grow the hair follicles over 1 millimeter within a week. And what we saw is that on day 7, however, the hair follicle cycle changed. So we see a detachment of the hair fiber of the bulb on day 7. But this is the time range, and that was nice to see. So the next step was to go and repeat this in a 3D environment. And after a couple of failures with different approaches, we figured out that to do that, we have still to maintain a good nutrition supply to our hair follicle, which is pivotal. So what we did after a couple of failures and other approaches is we used the technology of 3D printing. And what we did is we printed a PCL scaffold with pores in which we printed a sacrificial hydrogel, which prevented the following filling of a pre-polymerized collagen from leaking. The collagen is our substitute, our DIMEAS substitute. And then we fixed, before the collagen was polymerized, the isolated hair follicles into this sacrificial hydrogel. And then we polymerized the collagen at 37 degrees and simultaneously removed the sacrificial hydrogel. And by that, we created some kind of channels for direct nutrition supply, at least to the level of our hair follicle bulbs. And then we compared again the hair growth to our conventional culture. And we saw, okay, here in blue, the blue line is the 3D model, the orange line, the conventional method. And we saw, looks quite equal. So there was a nice progress. And we saw also, again, the same phenomenon in the 3D model on day 7, the detachment and the hair cycle change. But we have quite a big time range, at least from day 0 to day 5, 6, to make any kinds of experiments. So then we wanted to do a quick proof of concept. What we did is we took commercially available hydrophobic fluorospheres. It's important that they are hydrophobic, that they do not cross our collagen matrix of about 500 nanometer in size, and added that on top of the matrix for 24 hours. What we wanted to see is a follicular transport, of course. We first of all excluded that without any hair follicles, the particles cross our collagen matrix. And then we applied it on the hair follicle loaded model. And then after 24 hours, we saw a follicular transport of the fluorospheres. And then we wanted to see what kind of parameters really influence this transport. And what we did is we took an additional fluorosphere size, so 500 nanometer and now 200 nanometers, and reduced the exposition time from 24 hours to 3 hours. And then what we could see is, obviously, there seemed to be a, like also reported in literature, a size and time dependency of follicular transport. And this is what we are doing right now, and it's upcoming. We are really making studies to really characterize the follicular transport in this model, but this is still upcoming. The next thing to prove the concept was to see whether we can inflame and infect our model, that we can cure it afterwards in to validate the model somehow. And what we did is to inflame the model, we infected with a Staphylococcus aureus strain, GFP strain, by drop infection of our hair follicles. Here we can see without any hair follicles, they stick on the surface, and here with hair follicles. And then we wanted to see basically just how the bacteria enter our hair follicles. However, after the first couple of experiments, we saw always somehow the bacteria colonize around our hair follicles rather than going inside our hair follicle opening. And my primary hypothesis was maybe I somehow blocked my hair follicle opening by covering the opening with pre-polymerized collagen. So I really was careful with the handling and tried to prevent that. And then finally we saw, okay, we can see a nice colonization inside our hair follicles. And that was also nice progress. And now the aim is to see how the bacteria grow, and the growth behavior is more in detail. And also after a treatment. Now a quick conclusion. We established an isolation protocol. We cultured the hair follicles in a 3D environment. We also applied a quick and dirty proof of concept of hydrophobic particles which enter the hair follicles. We saw that bacteria can enter and colonize the hair follicles. And what is missing is that we really optimize the whole processing and design of the monomer. And we are currently doing a particle penetration study and how they are influenced by the parameters. We want to characterize the bacterial growth behavior and reaction to different treatments. And on the long term, we want to also add an epidermis substitute to go even to mimic more closely the real skin situation, also by adding maybe cells. And now I want to use the remaining time to talk shortly about another application of our 3D printing technology. Namely, we want to also 3D bioprint, as mentioned, bacterial biofilms, which we talked about in the previous part also. And as you know, these bacterial biofilms are a big problem. They can occur on basically everywhere, on medical devices, on all kinds of tissues, lung, which is also a big focus in our research group, skin, hair follicles, gut, and many more, in all kinds of shapes and dimensions. And there are promising novel anti-infectives to fight these bacterial biofilms. However, currently they are specially tested on animal models, which are basically infected with bacteria-loaded alginate beads. This is the current models which are very often used in science. However, there is a major drawback. These biofilms are not really growing like in vivo on the in vivo epithelial barrier. Now they are growing in the alginate beads. Other drawbacks, as mentioned before, are again genetic, immunological, and anatomical differences. Again, basically the same problems. So what we really desire, what we require are good in vitro alternatives. And ideally, we want to have co-cultures of human epithelial cells and bacterial biofilms to really mimic a biofilm chronic infection. However, the problem is when we just put planktonic bacteria on top of epithelial cells to form a biofilm, the cells die rapidly due to overgrowth of the bacteria. So nowadays, the research groups try to grow the biofilm separately and then transfer the ready biofilm on top of the cells. However, there you have the problem that you disrupt this biofilm, that you also, by the transfer, you do not really get reproducible biofilms on top of these epithelial cells. So that's why we came up with the idea, how about using 3D bioprinting and growing the biofilm in a hydrogel, so that we have a ready biofilm in a hydrogel, and then we print reproducibly the biofilm on top of the epithelial cells in any shape and dimension you want. You can also, however, you can also use the tool without any cells and just print the biofilms in any required shape you desire and require for any kind of antibiotic testing. And what we did to do that is we had a gelatin alginate hydrogel with inoculated bacteria, E. coli MG1655 as a biofilm model strain for now. And then we pulled the hydrogel with bacteria into a syringe and let the biofilm grow in the syringe. And after the biofilm grew, here on the piston here, we transferred the biofilm into a cartridge gently. And then we were ready to bioprint immediately. In any required shape we need it. You can also follow this by cross-linking steps for any kind of assays and experiments you want, with calcium chloride, for example. And we saw, okay, the bacteria here printed on agar plates survive the printing process. And when you then print any kind of structure, you can, for example, stain your biofilm matrix, that you really can observe your biofilm matrix, which is here the case. Here you can see the staining of the bacteria, the nucleic acid, and here the biofilm matrix stained, here the overlay, compared to planktonics. And also in the, you can see the biofilm pattern at the SEM microscope. And what you also want to then make sure that you basically kept your biofilm properties after bioprinting. And to see that, what we did is quickly a antibiotic susceptibility assay. We printed as well biofilms and planktonic bacteria and compared this for their susceptibility to two different antibiotics, here ampicillin and tetracycline. And we could see the bioprinted biofilms are still more resistant than the counterpart of planktonic bacteria. And an additional assurance that we are really sure that we have a biofilm state after printing is what we did is a metabolic profile analysis and compared it to the planktonic bacteria again, because we know there is a huge difference between the metabolic profile of planktonic and biofilms. And this is what we then also saw and confirmed. Here in this diagram, you see compared the planktonic bacteria in green and the bioprinted biofilm, they are quite different in their metabolic profile. And here in this diagram, you see the native biofilm, which is not printed, and the bioprinted biofilm, which is still pretty comparable, pretty similar in their metabolic profile. And I hope now I could give you a quick overview about how we use the 3D printing technology to, kind of design these in vitro models to fight against, as a platforms for testing nanoantibiotics against bacterial infections. And with that, I want to thank you for your attention. I want to thank all my supervisors, especially Professor Klaus-Michael Lehr. And if you have any questions, don't hesitate to ask.
So thank you very much, Sammy. I think very impressive results, and there will be a lot of discussions, especially maybe on your model, because, for example, you can not remove the follicles from the skin, but maybe use the skin as a kind of scaffold in addition. But we will discuss that later. And maybe you have seen that we have this debate room where we can join now. But I think it is very important that we make a short break, that you can stretch your legs and maybe drink some water. And I think we should meet in a very few minutes in this room. Thank you.